Good afternoon, everyone. Thank you for being here. It's a pleasure to have you back. We are about to enter into session 2 of the Water Data Symposium. It's a hybrid event this year. We started yesterday morning with our part 1 of the Water Data Symposium. And then we had a workshop in the afternoon dealing with the challenge questions and teams and data issues and lots of cool organic exploring. We reconvened the workshops this morning, had some more fantastic discussions, and teams are forming and projects are forming. Data is being discussed. And so now we're here to stagger that with some more presentations that are going to be really inspiring and help inform, I think, even additional ideas for how to go forward with this water data challenge and the work ahead of us in California. So thank you all for being here. I am Greg Gearheart. I am the Deputy Director for Information Management and Analysis at the State Water Board. I am co-convener of this with Anna Holder, and we also had some great help from others, Nick Martirano, Jake Pasner, some others, and all the helpers that are on the crew today. So big thanks to all of you and shout out. So next slide, please. I'm going to cover some logistics. And so first and foremost, as you are probably already aware, we're recording this and they will be posted. So that's a shortened link, and we'll provide that link later to everyone who's attended. Please submit your questions to the presenters and panelists using the Q&A window at the bottom of your screen. That is the only way to submit questions. So please do that. If you have technical issues, go ahead and email anna.holder@waterboards.ca.gov. And we will get back to you as soon as possible. Next slide. So this afternoon's agenda is primarily water quality related symposium presentations. We do have one more thing tomorrow, another workshop of the water data challenge type. So if folks are still kind of on the fence or if they have additional questions and they've already been to these workshops, feel free to drop into that workshop tomorrow morning. But today we're going to go from 1:30 to 4:30 with a solid block of really intriguing presentations. Next slide, please. Oh, and I forgot to mention the obvious maybe, but the salmon mascot this year was selected to sort of symbolically lead us into the future. We have a really good write-up on it in the program. So if you haven't looked at the salmon write-up in the program, I encourage you to look at that. Today's agenda, I'm sorry, back one, and I can do that too, I suppose. Today's agenda, we're going to have this plenary session with a presentation by Karen Mogus, who's Deputy Director of Water Quality at the State Water Board, strong partner, and also co-chair of the Water Quality Monitoring Council. There'll be a conversation around that, and then we'll have the session 2 Water Data Spotlight that's going to be looking at a lot of fascinating water quality related topics. And then the symposium wrap up and upcoming events is going to start at 4 o'clock. Next slide, please. So with that, I'd like to introduce Karen Mogus. Karen is Deputy Director of Water Quality at the State Water Board. Like I said, she's also co-chair of the Water Quality Monitoring Council. She has a long-standing relationship to the programs that I'm now overseeing, the swamp surface water ambient monitoring program, and the data engagement and the turning data into information at the water boards. I followed in her huge footsteps. And so it's with great pleasure that I'm here to introduce Karen to give us a speech.
A speech, hopefully as inspiring as you teed it up to be. Yeah, Greg already introduced me. I'm going to wear 2 hats today. My hat as the Deputy Director of the Division of Water Quality and really a data user. Well, I suppose my programs are both data users and data producers, as well as my hat as the co-chair of the Water Quality Monitoring Council, and try to inspire you all to think about why water data is important to your organization and how we could do better to collaborate. I feel a little bit like I'm preaching to the choir today, but hopefully I get a message out to some of you and make some change. So next slide, please. So everyone needs data, right? everyone, all kinds of types of data. And of course, we're talking about water data here. The state and regional boards have regulatory responsibility for protecting the water quality of nearly 1.6 million acres of lakes, 1.3 million acres of bays and estuaries, 211,000 miles of rivers and streams, and about 1,100 miles of exquisite California coastline. The state board also oversees the allocation of the state's water resources for diverse uses, such as agricultural irrigation, hydroelectric power generation, and municipal supply, to name a few. To do this, the water boards implement a wide range of water quality, water rights, and drinking water planning and permitting programs. And all of these programs rely on the best available science and data to support the critical decisions we make that affect human health and the environment. And climate change, as you see here in some of these photos, show increasing numbers of and frequency and intensity of wildfires causing debris flows. Here at the Ventura River ecosystem is one example. Increasing numbers of widespread toxic algae blooms, and then the constant stream of chemicals entering the marketplace exacerbate existing water quality and quantity problems, making innovation in data collection and assessment all the more important. Next slide, please. So everyone needs data, but no one has enough resources to monitor for everything everywhere. And that's why collaboration and the ability to combine data collected by different entities for different purposes is critical. And yet it is quite challenging. Next slide, please. So I'm going to give a couple of examples of why this data combination is challenging. Here's one. Under the Clean Water Act, states are required to assess their waters every 2 years, identify waters that are not attaining standards, and report those impaired waters to US EPA. The assessment is based on readily available data from water board programs, and we work really closely with Greg's group to collect that data and make it available for our assessments. And we also solicit those data from external agencies and entities. Prior to the establishment of California's listing policy in 2004, the 9 regional boards independently assessed their data to determine those that were impaired. And as a result, the assessment framework was not consistent across the regional boards. And as you can see from this map, it would appear that the waters in California's North Coast region are the worst in terms of water quality, when in fact, many could argue that many of the waters in that area are the most pristine in the state. So this demonstrates an agreed upon assessment framework for data from a variety of sources is necessary to tell an accurate story of water quality in the state. Next slide, please. Here's another example of the challenge of telling stories with water quality data. On the left is a map showing biological assessment scores in perennial streams across the state, which were selected probabilistically. In other words, selected in a way that allows for asking broad questions, how's the condition of our waters? And by the way, these data were collected by the Surface Water Ambient Monitoring Program under Greg's group. While many waters are impacted, as indicated by the red dots you see here, this map also shows many waters are still in good condition and supporting aquatic communities. Now on the right is a map showing toxicity data collected in waters across the state. First, note that the entire geography of the state is not represented as opposed to the map on the left. And by the way, the big open spaces in the map on the left mean that there aren't perennial streams. And so it's not that we didn't sample there, we just didn't have any water to sample. On the first note that most of the waters are in urban or waters that we sampled for toxicity are in urban or agricultural land use areas. And second, note that this map would lead one to believe that there are far more waters that are toxic to aquatic life than there are not. Yet the biological assessment that spans more waters across many land use areas shows that many more waters are in good condition than one would think if they just looked at the toxicity data. So the questions that drive monitoring program design also impact the story of water quality in California. Next slide, please. So what are we doing to address these issues to answer the important water quality questions? Well, first in 2006, the legislature established the California Water Quality Monitoring Council as a partnership between the California Natural Resources Agency and the California Environmental Protection Agency. The council mirrored and coordinated closely with the National Water Quality Monitoring Council and was established to maximize efficiency and effectiveness of existing water quality data collection and dissemination, and to ensure data is made available to decision makers and to the public. To achieve these goals, the council relied on multi-agency coordination and communication. They formed theme-specific work groups that developed portals to comprehensively assess available data and identify data gaps. And the council provided overarching guidance to the work groups. Next slide, please. And here's a screenshot of the various portals that were developed by the work groups under the auspices of the Monitoring Council. And as you can see, the portals are designed to answer questions that are important to the public, such as, is it safe to drink or swim in the water? Is it safe to eat fish caught in California's water bodies? And are our waters healthy enough to support ecosystems? The portals not only attempt to answer these questions, but also demonstrate where there are data gaps that make it difficult or impossible to answer the questions in certain areas. Next slide, please. So the council is one way the state is working to address water quality challenges, but not the only way. In 2016, the legislature passed AB 1755 to create an open and transparent water data platform for California that spans beyond water quality data. The Internet of Water Coalition is a group of organizations that work together with federal, state, and local government partners to guide foundational water data infrastructure across the nation and create a community of people and organizations using water data to make decisions with informed data. Oh, no, not quite. I wasn't done. And the California Open Data Portal was established to provide the public ready access to government data on a variety of topics, including water. The Water Board and the Monitoring Council partner with each of these efforts to achieve our open data goals and turning data into information. Next slide. So as I mentioned, the council has successfully established a number of work groups that have developed data portals to answer a variety of questions important to the public. I will note that the council has very little dedicated funding. Just one person is dedicated to the program and serves as the director, and his name's Nick Martirano, that many of you are already very familiar with. He gets everywhere when it comes to monitoring data. So all of their efforts have been done by volunteers with expertise and energy to put to the effort. The work done to date was intended to leverage funding for the council, but that hasn't materialized despite multiple efforts. Recognizing the funding is likely to remain an issue, in 2020, the council updated their strategy to make the most of the partnership and collaboration that have formed. The 2020 strategy builds on and recognizes the value of a decade of progress, establishes realistic goals and objectives to fit the available resources, and recognizes and calls for coordination with new water data initiatives. It also establishes 6 strategic goals, and on the next slide lists the 6 goals. And rather than talking about them right now, I'm going to go through each one and why they're important for my other role as the Deputy Director of the Division of Water Quality. So next slide, please. So how does the council support the Water Board and its goals? I'm going to walk through the council's strategic goals and give examples of how they support the Water Board's work, in the hopes that you will start to think about how the council might support your agency or organization too. Next slide. So the first goal is to assess the performance of water quality monitoring programs, which has a couple of components. First is to inventory the monitoring occurring in the state. This is important to the Water Board because we need data for our water quality assessments. We also collect data ourselves and want to ensure it's done efficiently and effectively and doesn't duplicate other efforts. The second component is about making sure the monitoring that is being done produces data that answer questions. Monitoring is expensive and resource intensive, So we cannot afford to collect data that can't be used to answer questions and make decisions. Next slide, please. The second goal builds on the first with a focus on the quality of the data collected and making sure it can be used for multiple purposes, not just for the purpose for which it was originally collected. Again, we can't afford to collect data that can only be used for one specific purpose. We need to be able to leverage data collected across organizations to answer the board's questions and support informed decision-making. And that is really the foundation of that is guidance and implementation of strong data quality principles, as well as methods that are comparable across monitoring programs. Next slide, please. The third goal relates to the need to be able to turn data into information. Interpretation thresholds are needed to tell us, what does the data mean? We have water quality objectives for some chemicals, but not all. We also have analytical tools that tell us there's a chemical present, but not whether it's harmful to the environment or to the public. And so without interpretation thresholds, the data lack meaning and ultimately cannot be used or transformed into useful information for decision makers. Next slide, please. The fourth goal recognizes the need to continuously improve our monitoring techniques as new methods and technologies become available. With new technologies comes the need to establish data quality and data management protocols. And coordinating this work early in the development of new methods minimizes the duplication of effort and ensures data collected across entities is usable for multiple purposes. A recent example of this is the development of the methods to monitor for SARS-CoV-2, the virus that causes COVID-19 in wastewater. Next slide, please. The fifth goal builds on all of the previous ones and is the ultimate purpose of collecting data in the first place. At least I hope it is. It's a commitment to turn data into useful information by supporting the work groups, leveraging published open data, building work group membership data analysis and visualization capacity, and prioritizing what information is most needed and by whom. Why collect data if we aren't going to assess it? Data that's collected and not utilized to answer questions or make decisions is a stranded investment that we cannot afford. Next slide, please. And finally, the sixth goal is to continue to build partnerships and foster collaboration to continuously improve how we collect data, how we store data, and how we turn it into information that helps us do the critical work to protect our water resources. And as I said at the beginning of my presentation, no one entity has the resources to monitor everywhere for everything. So we need to rely on the water data community to get the information we need to do our jobs effectively and efficiently. We owe it to all Californians. Next slide, please. So again, at the risk of being accused of preaching to the choir here at the 7th Annual Data Symposium, I challenge you all to think about why water data matters to you and your organization, and to think about ways you can contribute to the collaboration we need to be most effective now and in the future. And I think most of you have already made lots of strides in that direction. Otherwise, you probably wouldn't be here listening to this presentation and participating in the symposium as well. So thank you for inviting me to present. I hope I've inspired you a little bit today, and I'm happy to answer any questions if we have time.
Hi, Karen. We do have one question for you. Question is, how can we better highlight the need for thoughtful investment in monitoring programs to the legislature and other funders so that we can make more thoughtful decisions and regulations?
Really good question. I have spent the better part of my career trying to deliver that message. And in fact, my entire career, I have been trying to deliver the message that if you don't monitor it, you cannot manage it. And I don't know how to convince. I mean, the problem is that monitoring is a long-term endeavor that costs a lot of money. And so it's really difficult to justify a long-term monitoring program that increases over time, and we've got money that we get every year, but it hasn't increased, and therefore our ability to use it to monitor decreases every year. And so if I had that magic message, I would have already used it, and we'd have monitoring money flowing in. And so we attempt to take advantage of opportunities when we can to hitch our wagon to big ticket items. So most recently, wildfire impacts to water quality was one way we attempted to make the case that we need better and longer term data to make sure that we are answering the questions and prioritizing where we do our water quality work in a better way. And honestly, we have not been successful. So if any of you have brilliant ideas on how to tell that story, I'm all ears.
Thank you, Karen. That's the only question that's come up for you. So I will pass it to Greg.
Okay.
Thanks, Beth. And thank you, Karen. Thank you for coming and sharing your time with us and your expertise and the business end of the monitoring data at the Water Board for sure is in the water quality programs. So thank you. Next, we have a panel, and the panel will begin with a presentation about the SWAMP program, and I will introduce the speakers. They are two of my senior staff in the Office of Information Management and Analysis, Ali Dunn and Tessa Foget, both senior environmental scientists and both amazing. And we're honored here to have them to talk about SWAMP. So take it away.
Cool, thanks, Greg, and thank you, Anna, for having us here today to talk about SWAMP. We're going to focus on the data that we generate through our work. And I just wanted to mention to everyone, please also copy me and Tessa if you have brilliant ideas on monitoring funding that you'll be sending Karen, because we're interested in that. My name's Ali Dunn. I'm the SWAMP statewide coordinator, as Greg mentioned, and I'll be co-presenting today with my SWAMP colleague, Tessa.
Hi, I'm Tessa Foget, and I'm the SWAMP QA Quality Assurance Officer and Database Manager.
And we're going to kind of tag team things today. I'll kick things off with a brief overview of who we are. I'll talk a little bit about each of the statewide monitoring programs, and then Tessa will talk about this high standard that we hold our SWAMP data quality to, as well as how to access data. And then I will kind of take it home with a few examples of data dashboards that our team has developed just to kind of help improve data accessibility and interpretation and usability. And next slide, please. So I'm not sure that everybody is familiar with who is the SWAMP. So we were created over 20 years ago in response to some legislation that basically said, I think Karen said it earlier, like monitor everything everywhere. So this directive was to develop a comprehensive statewide surface water quality monitoring program to assess the condition of California's watersheds. But we can't be everywhere all the time at every water body for everything. So that's where the statewide programs come into play. There was a lot of strategizing and planning in the early days of SWAMP to kind of organize these statewide programs and generate some data that's useful to Californians and other water board programs. And we do that as this functional kind of collaboration between the water boards and our partner scientists. So this doesn't represent everybody we work with, but we kind of threw a splattering of logos of folks that we work closely with, including people at Fish and Wildlife, CSUs and universities. The list goes on. Next. Oh, you're on it. That's good. Next slide. So our work is guided by this vision and mission included here, which I won't read. In short, generating this high quality, accessible and usable data for informed decision-making and informed California communities is at the heart of our work and everyday efforts in SWAMP. Also central to our work is both the Water Board's open data resolution and the recently adopted racial equity resolution. So in recent years, the program has made these very intentional efforts to operationalize equity in our work and to ensure that all swamp data is made open and accessible. We've also focused efforts internally to better align and integrate our monitoring and data quality standards throughout related water board programs. Next slide, please. Okay, so I just want to spend a little bit of time talking about swamp statewide programs before I hand it off to Tessa. So because California is very big and complex and diverse, and I say we as though I was there, maybe Karen was a part of this, but we prioritize efforts to address a set of key management questions that focused on some core beneficial uses. So that's water board jargon, essentially for the resources or the services that California aquatic systems provide and that the water boards aim to protect and restore. We also focused on different representative water body types because we can't be everywhere all the time. And as a result, these statewide programs came out of that. And I'll talk about those in the next few slides. And then at each region, there's also a designated swamp coordinator, and they identify their monitoring priorities to answer or address specific regional management questions or water quality issues. So as an example, the Central Valley Water Board, the regional swamp program there, among many things, has a regular fecal monitoring program in the Michigan River. Next slide. Okay, so just to kind of start things off with the bioaccumulation program, this program focuses on the protection of human health by assessing fish and to some extent shellfish. We're trying to do more of that in the future, consumption, safety, and fishable waters. So you'll often kind of hear this program as the Safe to Eat program, since there's a direct link of data use in that way. So the data generated by this program are used by the water boards to assess the impairment of fish and shellfish through the integrated report process. And that process is essentially the water board's way of identifying whether a water body is in good shape or not. The data is also used by the Office of Environmental Health and Hazards Assessment, or WEHA, to support the development of fish consumption advisories. And that's kind of where that safe to eat question is being addressed. The data is also essential to the creation of a statewide mercury program. So I just wanted to go back to one thing I'd said earlier about SWAMP trying to be very intentional in our efforts to operationalize equity in our work. And I think this program is kind of a shining star example of that under the leadership of Anna Holder, who's also done a great job at organizing this symposium. And essentially what's happening there is the program is undergoing a realignment process. And through that process, we hope to guide future bioaccumulation monitoring so that it aligns with the public needs. So in particular, we're looking at areas where communities rely heavily on fishing for consumption, subsistence, sustenance, and cultural purposes. Right now, we're focused down in San Diego region. And Anna and Chad Laughlin and others put together an advisory committee, which includes both tribes, tribal advocacy groups, community-based organizations, and then some of us statey and federal agency folks. And the goal is to move efforts to other parts of the state to continue that realignment and make adaptations to address the public's needs. Next slide. Okay, so the bioassessment program focuses on protection of aquatic life beneficial uses by assessing biological indicators of ecosystem integrity in streams and rivers. So there's two core monitoring programs, a statewide probability-based survey. So if you remember the map that Karen showed with the colored dots, that data was used, or data from this PSA program was used to generate that map. And then there's also a targeted reference program. So the data and tools that come out of this program are used in several different applications, including the measurement of ecological integrity and identification of high quality water bodies. And this data has also been really essential to efforts to develop a watershed assessment dashboard, which has been a primary focus of the Healthy Watersheds Partnership, which is a monitoring council working group. And we're pretty excited about this dashboard because it's integrating biological data with other watershed related data sets that will provide a data-driven foundation for understanding watershed health statewide to support strategic decision-making, communicate potential management actions, including things like watershed planning and high quality water designations. At least that's the dream. Okay, next slide. Okay, so the freshwater harmful algal blooms program, this is one of our more public facing, you can go back one slide, I don't know if there's a lag, but the freshwater harmful algal bloom program, thank you, is more public facing. We go out and monitor kind of more in response to reports of blooms that we get from the public and from other agencies or tribes that have regular monitoring, and then use that data to help inform postings that go up at water bodies that say, don't go in, cyanotoxin levels are really high, keep your dog out of here. Or actually we went out and tested and it looks really bad, but toxin levels are low. So you could go in if you so choose. The program is just, has continued to grow since its inception several years ago. And like I said, data right now is primarily reactive, but we're hoping to adapt the program and develop a more statewide, comprehensive, proactive program in the near future. Next slide. Okay, and last but not least of the statewide is the Toxicology and Contaminants Program. This was previously referred to as the Stream Pollution Trends Program or SPOT program. And the focus here is on the protection of aquatic life beneficial uses. And we do that by characterizing toxicity and contaminant concentrations at the bottom of targeted watersheds throughout the state. We also conduct special studies, we do some screening surveys and other monitoring collaborations. I think one of the kind of big messages out of this program is some of the data that we've collected have documented toxicity associated with both existing and emerging pesticides. So that's an area we're really kind of honing in on and potentially focusing on in the future. And we do that with close collaboration with Department of Pesticide Regulation and Department of Toxic Substances Control, as well as folks in Karen's group in the Division of Water Quality. And with that, I'm going to turn it over to you, Tessa.
Great. Oh, there we go. So what makes swamp data useful to many people is that we have a commitment to quality assurance and planning, and we have dedicated staff to it, which means that swamp data are of known quality and any issues with the data are documented so people can decide if they're appropriate for their use based on that documentation. So we focus on making the data publicly accessible and develop resources and tools to help others use our data and also to plan your own monitoring projects. Next. So our overall program and each of the statewide programs Ali mentioned, as well as the regional monitoring programs, follow this adaptive project management process of plan, document, implement, reflect, and report. A key component of the planning and documentation steps of this process is the Swamp Quality Assurance Program Plan, which sets up basic requirements and processes for all swamp monitoring projects. So beyond the program plan, regional swamp monitoring projects do a short project write-up to describe their project in more detail. And it's an easy to follow series of questions that walks through the US EPA data quality objective planning process, which is often documented in a quality assurance project plan or QAP. And our statewide projects each have their own QAP that they use. So QAPs can be daunting to write for a lot of people. So we have developed a resource to help simplify it called the QAPs Made Easy worksheet or workbook that walks through those same series of questions to help other folks use that. So if you're writing a project plan, we encourage you to borrow quality control acceptance criteria or anything else that works from the SWAMP program plan because they are appropriate for many environmental monitoring projects. Next slide. So a foundational part of SWAMP project planning is identifying the intended data use. This will determine what type of quality control requirements and level of standardization is appropriate for the specific project. So a regulatory monitoring project would have stricter quality control requirements and use ELAP accredited labs and methods, whereas investigative project wouldn't need those strict requirements. Next. So you can find swamp data in the CEDN database, and flat files of CEDN data are also posted on the California Open Data Portal. SWAMP has also developed several data dashboards where you can interact with SWAMP data and download it from there. And Ali is going to show some of those dashboards and tools. Next slide, back to Ali.
Well, thanks, Tessa. And I'm going to kind of jam through these because I realize I don't think we have too much time, but I just wanted to say, you know, we've got over 20 years of extensive and high quality data sets that we've been using to tell these different stories about California water bodies and watersheds in a way that makes sense to a broad audience of data users. So we wanted to share with you just some of these dashboards that the SWAMP team has developed and that were created based on the user's needs or that are user-centered design. In all cases, the goal was to improve communication of environmental and human health conditions, like I said, to different audiences, depending on who's interested. And we also hope that they encourage collaboration and the use of SWAMP data, both within and outside the water boards. Next slide. So the SWAMP Data Dashboard is an interactive tool for exploring and downloading water quality data collected by SWAMP since the year 2000. It's interactive and provides a table for viewing, monitoring results and trends at the station level. This is the initial version. For all of these dashboards at the bottom of the slide, there's a small link so you can go play at your leisure and access the data there. But right now it's got data from CEDN and then additional SWAMP data sets, including tissue data and toxicity will be added over the next year. And if you go to that data tab at the far right, that's where you can learn more about the data used and how to download. And I just wanted to give credit to Michelle Tang. She's one of Swampawima's data unicorns that developed this and many of the dashboards that I'll be presenting. Next slide, please. And then at the regional level, there's tools that have been developed. In this case, this is summarizing data from the Lahontan Regional Water Board. I'm going to just keep jamming because time. And next slide. This is a more analyte-specific dashboard, again, developed by Michelle Tang. Not all the data here is SWAMP data, but some of it is. This Safe to Swim map is displaying bacterial sampling results for both coastal and inland monitoring locations. The data comes from CEDN. It's also accessed using an API on the California Open Data Portal, and you can download data again directly from this dashboard. And this guy is refreshed weekly. And next slide. So this is both a data dashboard and information hub. There's a lot of good stuff here, and I highly encourage you to go check it out. So this was developed by Peter Haupt on our team, and it's the Swamp Meta Barcoding Monitoring and Analysis Project, or CMAP. You can share information about your individual project here. You can see the data collected, find resources to help with your monitoring. There's illustrated instructions for sample collection, guides on best practices. It's super sharp, and I highly encourage you go check it out using that link on the bottom. And then the last one I wanted to share, next slide, is the HAB incident reports map. I think this is probably one of our most used dashboards because it's one of the primary ways to communicate where HABs are occurring and what their advisory levels are. I won't go into all the details of what the colors mean, but please, again, take some time, check out all of these dashboards. The data is all there. You can download and do your own thing with the data or use these as a way to interpret those results. And then last slide, please. Resources. Take a look. A lot of the links are here. There's lots of good stuff. And I'll put my email in the chat if you want to reach out with any questions.
Thank you, Ali and Tessa. I think we have time for one question.
Hi, yeah. We've actually got 4 questions, so we might have to deal with these in writing later on. But one of them here is, Given the limitations on resources and deprioritization of funding for monitoring by state-sponsored programs, how do you grapple with the prioritization and expenditure of resources? How often is that done? And how transparent is that process with stakeholders outside of SWAMP?
Want me to take that one, Tessa? I'll
Go
start
for
and then
it.
chime in. Okay. Great question. Well, I think you heard from Karen too, that this has been an ongoing challenge for statewide monitoring programs. And I don't know if I mentioned this, but we're constantly adapting and evolving, right? So what we've been trying to do in recent years, at least, and I can't speak to years before, but I think this realignment process is a good example of trying to involve tribes and tribal advocacy groups and community member groups from the start before we go out and monitor and ask, like, hey, does this make sense what we're doing here? And the regional boards too. Do these contaminants make any sense for you? Are we going to the places that are important to you? And then trying to adapt. And honestly, we're not getting more money. We're just sort of having to always shift funds around. So we always try to be open and in our decision making and transparent. But yeah, I think I covered it all. There were lots of questions in there.
Yes, there were. Anna has said that we can do one more, so I'll give you one that'll be a bit quick, a softball question here. Do any SWAMP programs collect data from state water project, water bodies, or cross-collaborate with DWR, since those are primarily surface water bodies, i.e. dams, YOLO pipe, bypass, etc.?
Let me take this one, Tessa. There you go.
Sure, go ahead. Yeah.
Okay. Well, I can say through the freshwater harmful algal bloom program that we do coordinate closely with them. There's lots of monitoring along the state water project in those reservoirs that are impacted by harmful algal blooms. So yes, there's one example of where we do team up. There's work in the Delta, which I think you'll hear about in the next few talks. So yeah, we do collaborate with them where it makes sense.
And before we sign off, I'm just going to quickly mention I am hiring 2 staff right now. I have an environmental toxicologist position and an environmental microbiologist, and I'll put those links in the chat again in case anyone is interested or to share.
Well, thank you both, Tessa and Allie. Great presentation, great story, the SWAMP program and the history and the future. So thanks. Our next speakers are Dr. Sam Beshevkin and Rosie Hartman, I believe. Maybe it's just Sam, but we've had the pleasure of getting to know them and working more closely with them in the data user working group through their efforts, and you're going to hear about that. So with that, take it away, Sam.
Great. Thank you, Greg, and thanks everyone for being here. Just waiting to have control. Okay, so as Greg said, I'm here to talk about a rich legacy of interagency collaboration, 5 decades of ecological monitoring in the San Francisco estuary. My name is Sam Beshevkin. I work at the Delta Science Program. I come from more of a data user end, but I work very closely with many of the data producers within the Interagency Ecological Program. So the Interagency Ecological Program, or IEP, is a consortium of 6 federal and 3 state agencies that informs management of the San Francisco estuary. They operate a lot of critical long-term ecological monitoring programs and also serve as a collaborative venue for estuarine science. Just to orient everyone, in case you're not familiar with the San Francisco estuary, I have a map of it on the right, and it's just the upstream portion from the Golden Gate up through the Sacramento-San Joaquin Delta. So I'm going to talk a little bit about the data that are collected by the IEP. There are 3 main, what I'm calling tools that are used to collect this data. There are boats, traps, and sondes. These traps are mostly fish traps of different sorts, and sondes refer to continuous monitors of water quality that that continuously collect data and send it back to the office. And the data are collected on a wide variety of parameters that include fish, zooplankton, phytoplankton, which includes harmful algal blooms or HABs, benthic organisms, water quality, flow, aquatic vegetation, and genetics. There are also a number of special studies that go on that use different tools and collect different parameters than listed here. So these samples are collected in the San Francisco estuary. You can see on the left a map of California with the estuary outlined by all the sample dots. And then on the right, I'm going to play a video showing how the spatial footprint of sampling has changed over time from 1959 to present. So you can see the footprint changing, expanding, and also the addition of different colors as new surveys have been added over time. They started with just one in 1959 up to probably 20 plus at this point. And this map does not represent every monitoring program that the IEP operates, just a subset. And as I mentioned, there's a number of different monitoring surveys, each of which has a different spatial footprint. So in this map, which just represents every sampling station in the estuary, each color represents a different monitoring survey. You can see that some colors are clustered in different areas, like the green is in Sassoon Marsh in the middle area, whereas others, like the red or the yellow, span much of the estuary. So when you're using these data, it's important to look at where exactly the data are collected. And when are the samples are collected is another important thing to know. So some surveys are year-round, others are just for specific seasons. Most of the boat-based surveys collect data at a monthly or twice-monthly frequency, while the SONs, as I mentioned earlier, collect data continuously. And the IEP has had an increasing commitment to open science in recent years. This includes actions such as interagency collaboration on QA/QC protocols to improve data quality, a huge focus on data publication, and the publishing of open source code on GitHub, and the IEP has its own GitHub organization. There's a great article that was published in San Francisco Estuary and Watershed Sciences a few years ago, Bearwald et al., that I'd encourage people to read to learn more about this. So by now, hopefully you're wondering how you can access these data, and I'm going to go into that in a bit of detail. The best resource is the IEP data website. And this website lists the locations where you can find data for all of the IEP monitoring surveys. You can see a snapshot of the table from that website at the bottom of this slide. And it also includes a ton of helpful information about each survey, like the type of data they collect, the contact information, and a description of the project. You can also access these data directly through data publications, integrated data sets, Shiny apps, and R packages. And I'm going to go into a few of these in a moment. So data publications are published on a variety of different portals. The most popular is likely the Environmental Data Initiative, which is a National Science Foundation funded data repository. But you can also find IEP data sets on the CNRA data portal, the California Natural Resources Agency data portal, as well as the California Department of Fish and Wildlife data website. The USGS has a data portal called ScienceBase. U.S. Fish and Wildlife Service posts data on their own website. And lastly, I want to remind everyone again that the IEP data website is the best place to find the locations of each, so you don't have to explore 5 different portals to find the data you want. For water quality data specifically, I'll talk about a couple integrated data sets that make access a bit easier. So the first is an integrated discrete water quality database. By discrete, I mean data collected by boat-based surveys. This data set is published on the Environmental Data Initiative, which I'll be referring to as EDI. It's also published on GitHub through a package for the R statistical programming language called Discrete WQ. The second database is an integrated continuous water temperature database. which has aggregated data from many of the sondes in the estuary, and that's also published on EDI. Zooplankton data are also available through an integrated database, which is as well published on EDI. It also has an R package on GitHub called Zooper, and there's an interactive online Shiny app that you can use to explore the data through different visualizations and download it to your computer to work on it if you're not a user of R. And you can find that Shiny app on the Delta Science Shiny Apps website. And lastly, fish data. Fish are what drive a lot of the management in this estuary. And we now have an integrated database of fish monitoring data that's published on EDI, available on GitHub through the R package Delta Fish. And there will be an interactive Shiny app coming soon to help access that. So I've mentioned Shiny apps a few times, and I just want to make a plug here that the Delta Science program has a Shiny apps homepage for apps relevant to the Sacramento-San Joaquin Delta, and we can host your Delta relevant Shiny apps as well. So if anyone out there is developing Shiny apps relevant to management of the Delta or the San Francisco estuary more broadly, we'd be happy to host it for you. So please contact us. We have an email, shiny@deltacouncil.ca.gov, or you can go to the homepage and look around there. So after I've talked about these data a little bit, you might be wondering why they're collected and how they're used. So I'm going to go into a few different uses of IEP data: real-time management, answering scientific and management questions, and in automated reporting. So for real-time management, I have two examples here. The first is the salmon monitoring team. And this team uses real-time data on juvenile salmonid abundance to decide when to close the Delta cross channel. And the Delta cross channel is a gate in the Delta that can be closed to keep salmon away from unfavorable central and south Delta habitats that might increase their mortality. The data are also used by the smelt monitoring team, which works on Delta and longfin smelt. They look at data on smelt abundance as well as water quality data as a proxy to understand when these species might be close to the huge export pumps of the State Water Project in the South Delta. And when these smelt are getting close to those pumps, they can actually recommend a reduction of Delta exports to reduce that danger. IEP data are also used to answer many different questions. So it's estimated that around 20 to 40 scientific publications come out each year using IEP data. And these publications can answer big ecological questions or local management questions, and they can take the form of peer-reviewed journal publications or technical reports. And there's really a huge volume of work coming out from these phenomenal data sets. And lastly, automated reporting is a new emerging use of IEP data. It's a great way to bring the data to decision makers as well as to the public as a communication tool and minimize the work of analysts by automating much of the work. So there are two main reports I'll just introduce here. The seasonal monitoring report is produced by IEP. It's on the IEP GitHub site. It provides an overview of many of the parameters in the San Francisco estuary over time at a seasonal basis. And similarly, there's a Delta smelt conditions report, which is produced by the Delta Science program. This is a bit more narrowly focused on conditions relevant to Delta smelt, but portrays some of the same parameters as the seasonal monitoring report. And that's available on my GitHub site. And that's all I had. So I wanted to thank Steve Culberson, the IEP lead scientist, was really helpful in drafting this presentation, as well as, of course, all of the IEP survey staff who do a tremendous amount of work collecting this really important set of data, without whom there'd be nothing to talk about today.
Thank you so much, Sam. We have time for a couple of questions. So, Beth?
Yeah, we do have a couple of questions. Sam, have you ever had an open source community member request a poll from any of your open source software? And what was that like?
So yeah, we definitely do collaborative open source software development within the IEP. As I mentioned, the IEP is not only a collector of monitoring data, but also a collaborative venue. And there's a lot of collaborations that arise from just the group of people involved in IEP. So many of the integrated databases I described are collaborations with other IEP members, and that collaboration has involved pull requests through GitHub and other types of interactions. I haven't received pull requests that were, I guess, unsolicited or not planned in advance, although I always welcome any contributions to any software I work on.
Okay, thank you. And then one more, and this is probably a fairly quick one for you. Are these data submitted to CDEN?
That's a good question. I don't think so. My experience with CDEN has mostly been trying to find toxicity data. I believe that the regional monitoring program might submit data to CDEN, but I think that's operated by the San Francisco Estuary Institute. I'm not sure if any IEP data gets on CDEN. I'm also not sure that there's really a clear line on what's IEP data and what's not IEP data, because there's a lot of people who work in IEP and also collect other data that is kind of a gray line. So I'd say probably not. Most of it is in the databases I mentioned. I guess one I forgot to mention is CDEC ending in a C, not an N, where most of the continuous sonde data is hosted.
Okay, thank you. And we had one more question that just came up. If someone wants to collaborate with the IEP, how would you recommend they do that?
Yeah, that's a great question. I think a really good starting place is the project work teams, which are groups of people organized around subject areas who meet at a monthly or quarterly basis to just talk about different topics like, regional fishes or zooplankton or estuarine ecology, phytoplankton, water quality. So that's a really great place to join up, and those meetings are open to the public, so anyone can join. And that would be a good place if you have a specific interest to interact with other people involved in it. And a secondary way would be just to send an email to anyone involved in IEP, and they'd be happy to connect you to the right people. And I neglected to mention too that our contact info is on the slide here, and I'd encourage anyone to email me or Rosie if you have any questions about this.
Okay, thank you. And Anna has just added the link for the project work teams to the chat. So that's available if anyone wants to look into that.
Thank you,
And that's
Beth.
all we have.
Thank you, Sam. Thank you for your presentation and your ongoing leadership in the world of water data collaborating. And I wish you the best, and I look forward to continuing to work with you. So thank you.
Yeah, thank you.
Okay, so our next speakers are gonna talk trash with us. Surprise, surprise. But Walter and Wynn are amazing partners, very, very generous collaborators over the years on developing out a much better trash data ecosystem. So Walter Yu works for Caltrans, and Wynn Calgar works for the Moore Institute, and they have a great presentation and some great tools to talk about. Thank you for being here.
All right. With that, we'll just go ahead and call out the slides, right? We're not running it.
It's your choice. You can either advance slides with your words or take control if they have given you that.
Okay. Wynn, how about we just call it out? Does it sound good to you?
Sounds good.
Okay. Yeah. So with that, good afternoon, everyone. Walter Yu with Caltrans, along here with Wynn Calgar, and here to talk about two projects we've been working on on the trash and microplastic data space. So with that, next slide, please. So again, here's basically the first project that we're going to present. It's called Trash AI. So the URL is pretty simple. It's trashai.org. This is going to be a case study and overview of a open source project, which was a collaboration between my volunteer work with Code for America, technically the Sacramento Brigade and Open Fresno, along with Wynn and the Moore, who works with obviously the Moore Institute. And so this project, as you see on the right-hand side there, thank you for the link there, Anna, is basically a tool to help researchers and users upload litter images and then using computer vision, identify the pieces of litter in images. So this would be very good for litter assessments, litter pickups. Basically, if you visit the URL there, you could batch upload your photos and you'll return results similar to what you see on the right-hand side. So with that, next slide, please. So again, I've introduced it. So that's the application. Again, it's a web-based application, and the audience is for scientific researchers, municipalities, and citizen scientists. We're really trying to lower the barrier to entry for leveraging computer vision. One thing that Wynn and I found, as well as the trash mining work group, and we do a lot of collaboration with the water boards, is that computer vision is not an entry-level topic. It does require quite a lot to spin up these technologies, maintain them over time, and then also interpret the results. So we've abstracted that. We've put in a lot of hard work to train the model, and then also develop a very easy, intuitive front end. So as you can see there, just in the top half there, but basically upload your photos, it'll train it in the web browser, and it'll return the results, and then you can download the results. And that's pretty much it. We tried to abstract away a lot of the details and really let the AI model do all the predictions and training. And again, we found that it was based on the feedback and refining over a couple of iterations that this was very easy and approachable. And then again, as I mentioned, this was a collaboration between the Moore Institute for Plastic Pollution Research and also, again, our Code for America Brigade, so Code for Sacramento and Open Fresno. There was a team of about 3 of us on the coding side, so to speak, to develop this application. Next slide, please. So we started out, again, given that was the, the finished product, if you will, we started out with a very modest, or actually very aggressive or very lofty goal of using computer vision and applying it to photos and doing a batch uploading and batch getting our results all back within one application. So in that regard, it was actually pretty lofty, but we started out with a very clear goal in mind. So knowing that trash in the environment is very difficult to measure, current standards are, as you see there on the right-hand side, is we would have human assessors go out there. At Caltrans, we do a driving methodology, which is, again, the same thing of having human assessors. It's subjective. It could be subject to human error, right? Two human assessors could disagree on what they see. So what we're hoping to do, not only, as far as assessments, but just in general with looking for litter in photos, is to make it more objective. And so one of the ways that this is being addressed by researchers and scientists is to leverage technology such as computer vision. And we used it in this case for objects that are somewhat up close, within say 20, 30 feet, but there's also different vantage points. There could be litter in drone imagery and sweeper imagery. So there's different vantage points, different types of objects as well. One of the things that we were looking at was not only to identify the litter, but you can see very closely in the photos that there's a label attached to those items as well. So it's a twofold problem of identifying where the object is in the photo, as well as correctly labeling it. So I'll talk a bit about that when we get into the model portion of this presentation. But this is kind of the problem that we started out with, again, is automating and making the assessments, human assessment of litter, more objective. And then the solution came up with was, again, to leverage technology, both in terms of AI, but also wrapping this up in a web application so that it's accessible to all, which we found to be one of the most requested items. Was whenever we presented, myself included, we presented at Casca, a different case study from Caltrans. A lot of the feedback was around, how do we use this? Where can we find this? So this, I think, scratches that itch. Next slide, please. So project development, getting a little bit into the technical details, just so again, it's not all just magic. So we developed it in 3 parts. I'll try to stay jargon free here for, not all of us are coders per se. So we developed the front end in a language called JavaScript. So that's what you see when you go to the UI or the URL there is you'll see a very polished, very easy to use UI, just upload, download your results. We created a backend with the Python programming language. So that's what basically holds the AI model to make the predictions of images, identify where, again, the litter objects are and what they are itself. And then we also had a tool set where we could, stitch everything together. So we brought the model into the application, and then there'd be communication back and forth between this, say, UI, where the user puts in the photos, and then we have the back end of the model to analyze the photos and return those results. So that's what we mean by the tool set and the communication there. Second bullet there again is very easy UI. So if you're on the website, you just click on that menu hamburger, pull down, upload your images, and then basically the browser does the rest. The application will make the predictions. It returns the results to what you see there. Again, as you would see there on the right-hand side. And then a little bit about the storage. So we store everything using Amazon Web Services. So if you have your trashy images, test them out, see how we do. It's not perfect. We definitely have a lot more work to do to refine the model, but we feel it's a very good first concept for first proof of concept. And then again, it'll support batch upload, which is a really nice thing to have. Instead of, if you had a couple hundred photos, you don't have to do them one by one. You could just throw the whole thing in there. It may take a little while because it needs to spin up and run in the browser, but in the end, it does return the results with the photos, which you can download, and then also the metadata to show where these objects are within the photo and what the objects actually are. Next slide, please. So a bit there about the AI model. So just the kind of the evolution of it. It didn't come in one piece prepackaged, so we did have to do some digging. So one of the first things we did was we started out with a very general model. So this is a model based on the Python PyTorch model framework, and it's called YOLO, not you only look once, not live once. Just a pun on that. But basically, it's a very general model trained on a couple 100,000 images. So it'll recognize everything from animals to cars, but that's not very useful in looking for litter. So what we did was then we took another data set called the Taco data set. So this is a trash annotation image data set. And we brought those into the, thanks for the link there as well, into the YOLO model. So if you think about it, kind of specializing this model, if you will, to then go look for litter. So it's generally enough to identify objects in a photo, but more so to actually then identify litter using the Taco data set. And then that process of doing that is called transfer learning. So we did that so that we could specialize this model and then bring it again into the application. Next slide, please. So future goals. So as I mentioned, we still have a lot of work to do. Even as you can see in the right side of the photo there was some litter that's not picked up. Some of the labels are mislabeled. So again, more work to do. One of the first things is we've open sourced this code. It's out there on GitHub. We'll be sure to share the link to the repository. We're always looking for contributors. Again, there's, especially on the computer vision side, there's quite a lot going on, but always happy to chat. We continue to work on this in our brigade meetings. And as I mentioned, increasing the accuracy of the labels and the classification, really just improving that model so that it can find these objects more accurately and then also label them correctly. And then also over time, as we'd like to allow users to annotate the images. And then if we do that, we can collect those images and contribute back to the Taco data set. That's one thing we'd like to do. And then at the same time, also then expand those annotations, such as the trash taxonomy that Win has also worked on. So those are, again, some of those goals is, again, improve that model, contribute to open source, and then also allow others that are interested in litter, data, and computer vision to contribute to the project. And I think that is my last slide. Let's see. Next slide, please. There we go. And Win, I will hand it over to you.
Thank you. So we are going to switch gears a little bit and talk about microplastics in drinking water. This is an up and coming concern for the public. They want to know, are there microplastics in our drinking water? We have found microplastics in drinking water, not within California, but in other states. And next slide. I'd just like to acknowledge my collaborators in funding. I'll go to the next slide for time. The Environmental Lab Accreditation Program is developing a new accreditation procedure for monitoring for microplastics in drinking water. And they're going to be monitoring starting this year and collecting some data. You can go to the next slide. And this data is really complicated. I mean, it is multi-dimensional. You look at particle sizes, shapes, polymer compositions. There are some really large data sets involved in doing the analysis. And so it really requires that we create some new types of database developments and visualizations. You can go to the next slide. And state policies require that new software be made open source and that data be made open. You can go to the next slide. So we are proposing that we are going to develop a new open source microplastic data portal where state accredited labs can submit their data. We're going to leverage several other open source projects that we have developed, OpenSpeccy, Tomix, Trash Taxonomy, Plastiverse. We've really been developing out an ecosystem of open source software for plastic pollution research and data analysis. And then that data would go from the microplastics data portal and be submitted to CDIN and SDWIS, which are the state-run data portals that would also like to host this data. It'll be reformatted into their format. Go to the next slide. This is our timeline. Currently, this project is going to go for the next 2 years. And we're currently in the stakeholder input phase of the project. We're going to transition into developing a minimum viable product and do beta testing. There will be tons of opportunities for people to get involved, both in developing out the code or providing feedback, or just letting people know that we're developing this project that's going to be totally open source, and seeing how best we can collaborate with others. We're also doing research as part of this, so there will be policy research on open source policy. How exactly do government agencies make an open source data portal, for example? That's kind of one big question I'm excited about. And then there'll also be a software manuscript. You can go to the next slide. You can go to the next slide. Thank you. So we have been a part of the Water Data Challenge for the past 2 days. Some of you have joined us, and we'll be there tomorrow too, in case anybody is interested in getting involved in this project or developing out a data challenge project associated with it. I think we have one more slide, and that's just our contact info. So thank you very much, and looking forward to chatting after.
Thank you, Wynn and Walter. As usual, very inspirational and advancing the front edge of leading data science and water data science. So thank you. Bev, do we have any questions?
We do have a couple of questions. First one, this is such an awesome example of leveraging data science to help inform and solve monitoring issues. What would you all say is the special sauce that made this a success story? Is it that trash is so easily relatable to non-water data folks, or is there something more?
Wynn, you want to go first?
Sure. Yeah. I mean, yeah, trash is very accessible to the public. I think the public is really excited about solving the trash problem. They feel like, when you go on a cleanup, for example, you feel like you can take matters into your own hands. It's a little bit more nuanced when you're thinking about other pollutants. So we like to call it in the trash world the gateway drug to all of the other environmental problems, because the average person really understands it and sees that it's a problem and they want to do something about it. And then you go, oh yeah, and like climate change, that's basically like, another part of the equation of how we're going to make this world a livable environment for us all. So yeah.
I'll also chime in as well, and I agree with Wynn. I think trash is very tangible in the world, in California, and I know that at Caltrans, especially whenever I mention litter, and what we're doing, there's a lot of feedback and everybody is very engaged. So I think it gets people very passionate and motivated about addressing the problem. So I think this goes a long way. I think also from the computer vision aspect, I get the easy part because it's in many ways the bright shiny thing in data science, right? What could be better than using computers to identify and analyze images, right? And then on top of that, we were very deliberate in the way we designed a UI to make it very easy to use. Really featured that photo, and I think I think with each of the demo, once I show it of Trash AI, people get it immediately. And so all I have to say is, oh yeah, we're just using AI to look at for litter, and I think the rest kind of takes care of itself. So in many ways, the job's been easy, you know, compared to say other balloons are just as important, but I think just litter is just more tangible. So back to the group.
Cool. I would have to agree with that. I mean, it's hard to get people excited about some of the constituents that get monitored because they can't see it. Their water still looks like water, but trash they can see and they can understand what's going on there. One more question. Does the Trash AI recognize trash not on streets, such as trash floating on lakes and rivers? Does it take a close-up of smaller trash? So two-part.
Sure. So the first part, in terms of not being on a street, yes. And actually, I encourage you to try it out. Get some photos of litter. I believe that Taco Dataset has some examples on their homepage, but try it in different settings. See how it does. You know, it should not the street or whatever the background is shouldn't really affect the what we call the you know, basically the borders of it, right? Or what they call edge detection, like actually identifying a litter object. So no, it doesn't have to be on a street or any given surface. And the only thing might be just the context, as I mentioned. If it's drone imagery or aerial or anything like that, it might be a bit harder to detect. And then what was the second question again, Beverly? Or the second part of that question, or I don't know who was asking. Sorry.
Yeah, that was me. Does the AI take a close-up of smaller trash?
That is probably one thing that, as I mentioned, yeah, like when it's drones, it gets almost into it starts to, because it was trained on Taco, where the objects were relatively within a reasonable range. Something, again, you would probably capture on a mobile camera. But say if you took it from a drone, the objects actually get very, very small. And then at that point, it probably requires some additional training or retraining of the model, which has been done. So say Tony Hale with SFEI actually did a project on that, of actually identifying just cigarette butts in the bay using drone imagery, but that requires a different data set. So in that regard, the project is open source. We've open sourced basically the base model. So if anyone was interested, and we did have some discussion in the morning session, some folks that are interested in doing their own version of the model, you can certainly clone and take the notebooks that we have in there, and then put in your own images, your own data set. And then there's also code in there to import, like the way we did imported Taco data, you could then import your own data set. So there is a way to do it, but as it is right now, it's basically, again, within that kind of mobile cell range. So.
Cool. We have one more question that came up. You mentioned being part of the Water Data Challenge this year. Are you looking for more people to join or support your projects? And if so, how would you recommend they connect with you?
I'll speak for myself and I'll hand it over to Wynn. Yes, most definitely. We talked a bit about that, but feel free to reach out to Wynn and myself by email. We're actively looking for Trash AI contributers, especially on improving the model. So for folks that want to, or even just learn, we're happy to share the code, do walkthroughs. I'll also be sharing the link with our Code for America or Code for Sacramento Brigade meetings. We meet every Wednesday in the evening, and folks can drop by if they're curious about the project. Happy to do demos, talk more about it, or if they have their own project related to trash data, I'm happy to talk about that as well and have office hours. So those are some different ways to connect with myself. And then Wynn, how about you?
Yeah, my email address is up on the screen right now, and we are definitely looking for people who are coders in any language, who are artists, who would like to make documentation or methodologies, or people who like to think about policies or do research on policies. So lots of opportunities to collaborate with us.
Great. And one more thing came up. Anna put a link into the chat if you have not seen it, linking to tomorrow's Water Data Challenge workshop, where if you are interested, you can connect with Walter and Wynn. So that link is available in the chat. Thank you, Anna. And that's it for our questions for these two.
Thank you, Bev, and thank you, Walter and Wynn, for again contributing your knowledge and sharing with us. And good luck getting people for your projects and your teams. So our next speaker is Emily Richardson. She works for the U.S. Geological Survey, and she's going to talk about the timeline of the Bay Delta history.
Thanks, Greg. Let me see if I can get control over the screen. Okay, here we go. Well, good afternoon, everyone. I will be sharing a tool that I have created with a group of folks from USGS and DWR. It's an interactive timeline application which showcases major Bay Delta events that are relevant to the science and management of the system. And there's really something for everyone in there, so I encourage folks to go play around with it. My name is Emily Richardson. I'm sitting on Nisenan and Miwok land, and I am primarily a water quality researcher with the USGS California Water Science Center's Biogeochemistry group, kind of a mouthful. I'm part of a team that collects a host of water quality parameters across the Bay and Delta at high spatial and temporal resolutions. Something that I have learned over time diving into and visualizing these data that we collect is that there are a substantial amount of factors that influence any given measurement. For instance, if you look at this data set here, which displays turbidity on the left-hand axis represented by these blue lines in both of the figures, with nitrate on top in gray and dissolved organic matter on the bottom in red, you can start to see some patterns forming. You can see that turbidity and nitrate have a very positive relationship. When one goes up, the other one tends to go up as well, and vice versa. Whereas with dissolved organic matter and turbidity in this data set, they seem to have more of a negative relationship, right? So they're moving in opposite directions here. But the relationships go far beyond these simple ones, right? Nitrate, turbidity, and dissolved organic matter may influence each other, but at the same time, they're being influenced by a ton of other factors. Maybe there was construction happening nearby, which affected the turbidity. Maybe a levee breached and flooded lots of agricultural runoff into the system, affecting nitrate. Maybe a wetland restoration project nearby was influencing the dissolved organic matter. All of these events and more impact our environment, and it's really difficult to keep track of every influential component. Say if in 20 years, an emerging new scientist is looking into long-term water quality trends in this area, they might not know of these events to take into consideration. We have stockpiles of data that increase by the day. We have no current compendium of major events that influence our data. So that's why we started this timeline project and why we think it's important. We also think that it's important to capture events that are not just relevant to water quality folks like me, but also to fisheries biologists, wetland ecologists, zooplankton researchers, managers, anyone who's interested in the science of the Bay Delta. That's who our target audience is. We would also like this to be a community collaboration. So the first effort to get our Bay Delta science community involved in this was to interview cross-agency retiring staff to capture events that might have otherwise been lost or forgotten about. And I think that's a really neat component to this project. We would also like this to be we're now soliciting feedback from users like you, and I will go over that more towards the end of this presentation. Okay, let's jump into the timeline. Here is your home page. Here you can go to get some good about information, some nice tips and tricks, and importantly, you have these 3 navigational buttons at the top of this page here. The first button is to enter the timeline. The second button is to enter a water year type specific timeline, and then the third is to a feedback form. Now, any page you can click on in this timeline, you will have your navigational buttons present and accounted for, including one to get right back to this home page. So it's very easy to navigate around. Okay, let's enter the timeline. Here's your default view. You can see that there are a few filtering capabilities. First, we have this category filter on the upper left-hand side. If you know exactly what you are looking for, you can come in here and uncheck the things that you don't want to see, click the apply button, and then it's filtered down to the timeline and the map. If you want everything back, click the all button at the very top, click apply, and everything returns. The current categories that we have are biological opinions, the establishment of different entities, different habitat milestones like pelagic organism decline. We have hatcheries information, infrastructure projects, invasive species introductions, legislation, levee breaches, and that includes the final floodings of certain islands and tracts where the levees were never repaired, and then also some other influential ones. We have species listings, monitoring program initiations, restoration projects, and then finally, the water year types for Sacramento and San Joaquin valleys. Up at the top in the center in the red box, you can see the decade slider. That is how you're going to navigate through time in this timeline. You can take the dot over here and slide it to any decade you want. This little decade number in this box will update so you know where you're landing, or you can slide one by one with these back and forth arrows. We have events going back 100 years to the 1920s. I will go ahead and show you what events we have from the 20s. Here we have all of our water year types, and then we have the final flooding of Lower Sherman Island and Big Break. We also have the creation of the Stockton Deepwater Ship Channel in this view, and if you hover over the events, you'll get a pop-up box that gives you some really good about information, some links to click on, related image, and some date information. If any of the events are associated with a place, if they can be found on a map, you will see them here in the map. And if you hover over the points, you'll see what they represent, where like what's happening in this area. You can zoom in and out of this map. You can pan around like it's a normal Google map. If you hover over counties that surround the Bay and Delta, you will see population density information. This is something that I think is really important. It's important for us to start really thinking about adding human dimensions to our research. So this is one very, very small example of a human dimension is population density. So I like to have that in here as a reminder. If it's cluttering your view, once you get towards the current decades where population is really booming, the shading of these counties gets really dark. You can come here to this layer control panel and remove the population layer if you so choose. But these are also really fun to play with. If you hover over San Francisco in the 20s, you'll see that there were already almost 11,000 people per square mile. That's a lot. Compare that to Sacramento County's 92 people per square mile, which seems a lot more comfortable to me. If you zoom to the current decade, you will see now San Francisco has about 19,000 people per square mile, whereas Sacramento has jumped up to about 1,600 people. So big changes and things you may want to consider. You'll see that water year type has a pretty big footprint on this timeline. I'm going to go back one decade to the 2010s so you can see kind of a full set of water year types here. You'll see them all laid out by what the water year type was. If you hover over these, you will get some runoff totals for the water year. However, if you want more detailed information, you can click on this water year type timeline, and it'll take you to these views here. These figures are depicting the running sum of mean daily flow by water year. The plot on the left-hand side is for the Sacramento River, and that's colored by Sacramento Valley's water year type, whereas this plot to the right is the San Joaquin River, and it's colored by the San Joaquin Valley's water year type. I'm gonna actually skip these next two slides for the sake of time, but I do want to give you a little bit of eye candy before I move away from these. If you scroll all the way to the left on your decade slider, oops, oh yeah, I'm in the right spot. You will get every single event put all together, which I think is a really beautiful image. If you hover over any of these lines, you'll see that it's actually a series of points that make up the line. So we have a point for every single day that was included in this CNRA data set. When you hover over, a pop-up box will show, and it'll show you the water year type, the water year, what the date was, what the running sum of mean daily flow was on that specific day. There is a bit of a typo here, but that has been fixed in the timeline. Okay, next I want to show you that this decade slider is directly tied to the timeline. So any decade you land on here will show right up back in that timeline. It's kind of working behind the scenes to synchronize them. So since I have scrolled all the way to the left, got all of my data plotted together, when I go back to the timeline, everything here will show as well. It's a lot of information, so you'll have to scroll down to see it all. But if you want to get to this view just by using this timeline page, you can also just similarly scroll all the way to the left to show everything all together in one place. Now, this is one of the tips that is mentioned on that homepage there. So if you forget, you can always go reference it or shoot me an email. I'm happy to answer questions. Okay, next we have the feedback form. This is where you can go to tell me any type of feedback you'd like to leave. You thought of an event that would be a good addition. You spot something that I should fix. You thought of a cool new feature, I'm all ears, or maybe you have just any other request. You want an entire new category added. You want, fires really impact your data, so you want to add fire data. Go ahead and tell me that here. I actually have, as of Monday, added fire data in, so go ahead and go play with that in the timeline, and you'll go see a lot of major fires. It's the top 20 biggest and the top 20 most destructive fires in California's history within the past 100 years. Okay, a few extra things I wanted to show. This is kind of a view zoomed out a bit, so you see this blue bar at the bottom. I like to view it in full screen mode, and I click this button on the right that is the full screen mode button. There's a lot of information here, and I don't want it to get smushed at all, so I like to view it as big as possible. We also, to the left of this blue bar, have the undo button, the redo button, and the revert to default settings button. And that will really just undo and redo any filters that you've added. And a cool new feature, even if you are really familiar with Tableau Server, which is what this dashboard is hosted on, you may not know about this new feature, which is one that I really, really am glad that they added and I really like. It is a download data button right here. You can click this. You can say you want to download the data. And here's the new part. You can come in here and you can right click any of these columns to remove them if you don't want that on your data download. You can click show fields and see if there's any behind the scenes columns that you want to add in. Some of these events have exact dates, and so those are kind of hidden from the view, but if you want to bring those in, you can do that here. And then finally you click download, and then it downloads to your computer as an Excel file. And that wraps up the features that I wanted to share. I hope this tool is really useful to you. I hope you feel welcome to send me any feedback or requests. Please, you're all welcome to do that. And ultimately, I hope you all have fun playing with it, because I certainly have.
That's fantastic. What a cool tool, Emily.
Thank you.
I have a question. So I'm curious how you did the data collection for all these events. And then a second part would be, how did you decide upon a data model to organize it? Sure.
So the bulk of the data collection was done by DWR, by Sadie Chomley. But I think a really big part of me wanting to agree to do this was when I first started here about a little over 2 years ago. I was tasked with finding all of the final floodings of all the islands that were never repaired, and that was roughly impossible to do. So that's why that is added in there, and kind of just like a use case of like, I can't find all of these events. And so just kind of narrowing it down what we want to add in that way. Sadie interviewed a bunch of retiring staff, and I think that's where a lot of these came from as well. And then the data model, I just kind of stuck it in a Postgres database. And so anytime I have a new event, I just append it to the table and bam, pops right back up in Tableau server. A really instant connection.
That's interesting. I was wondering if this is like a data type that is used in other, instances, maybe environmental work across the country or other things. So I wasn't like, this is new data to me. And so I'm always just curious, like, how's it organized and who decided to organize it? Do you have plans to expand, like go deeper? Like, can you relate granular information around events back into the, like, if you want to know the actual details of the flood or the water quality results associated with this event, those kinds of things. Are you thinking that way?
Actually, yes, we recently got funded to create a similar dashboard, but it would have long-term hydrologic and climatic data associated with it. And that is going to go back. I'm not entirely sure what our cutoff year for that is, but they're going to be linked. We're not entirely sure how we're going to link them. But we are working on a dashboard of long-term hydroclimate data as well.
Yeah, no, that's fantastic. Do we have any other questions, Bev?
We don't have any other questions in the Q&A or the chat, but I want to reiterate what Greg said about this being a really cool tool. Thank you.
Yeah, I could see this being really interesting to lots of people doing lots of research out there. So as usual, you and your colleagues there at USGS are doing really cool stuff. So thanks. All right, so we're gonna be a little ahead of time. Our next speaker is Dr. Laurel Larson. It's a pleasure to always see you again and work with you. And in fact, Dr. Larson and I met at a data challenge event in UC Berkeley's campus many years ago. So thank you for coming to speak and talk to us about the Delta Science Program.
Thanks, Greg. I was going to mention that. I am excited to have this chance to present to this audience. You all bring so much energy and passion and a diversity of experiences and training to the table, and it's just really exciting to have this opportunity. I also wanted to extend a congratulations to Emily. I heard about this tool when it was in the making, and it's so neat to see the success that you've had in pulling it together. So greetings, everyone. I come to you today as the Delta lead scientist housed within the Delta Stewardship Council. I also hold a position as an associate professor at UC Berkeley. And today I'm really going to be following up on what Dr. Sam Bashevkin presented earlier with respect to the IEP, but focusing more on the Delta Science Program. The Delta Science Program is a division of the Delta Stewardship Council, and like the IEP, the Delta Stewardship Council is really a science boundary organization within the state. We coordinate across many of the agencies and stakeholders performing science within the Delta. And the Stewardship Council itself is tasked with the mandate of managing the Delta for the co-equal goals of maintaining a reliable water supply and restoring, enhancing, and protecting Delta ecosystems. This is something that's of importance not just to the region of the Delta, but to the whole state. The Delta supplies 2/3 of Californians with fresh water, and it irrigates 40% of all the fruits and vegetables grown in the US. So this really is an area of critical national importance. So today I'm really going to be spotlighting the science program's role in data synthesis and highlight some resources and opportunities that I hope will be useful to you all as you put together your projects. Let me just try to advance here. There we go. Okay, so I wanted just to start out with the mission of the Delta Science Program in order to provide a broad overview. The mission is given to us by the 2009 Delta Reform Act, which is part of the California Water Code. And it states that the mission of the science program is to provide the best possible unbiased scientific information for water and environmental decision-making in the Bay Delta system. And it goes on to stipulate that this mission should be carried out through what has become our core functions: funding research, synthesizing and communicating scientific information, performing independent scientific peer review, and then coordinating with Delta agencies that promote science-based adaptive management. And today I'm really going to be focusing on this synthesis component. Now, synthesis at the science program takes several different flavors. There's synthesis that you could do of the literature. And one of the things that we put out is a periodic State of the Bay Delta Science, which is a synthesis of all of those 20 to 40 plus papers that Sam was talking about that come out per year, to really provide an understanding of what the state of the science is with respect to particular topics. And this year we have an issue coming out that is focused on primary producers in the Delta. Another thing that we do is we facilitate collaborative synthesis working groups. And my next slide will highlight one of those. We've done this through an ongoing partnership with the National Center for Ecological Analysis and Synthesis. And so our mission here is to provide training resources to the broader community to imbue them with data science skills and skills related to open science and best practices. And those working groups, and there's also a working group component that focuses on addressing big questions of management relevance. So I'll get into that in the next slide. But then the final thing that we do in synthesis is we provide virtual platforms for data integration and collaboration. And really, we provide a lot of resources to the community. We're working on providing cloud computing services. Sam mentioned our Shiny apps, and I'll show those again in a few slides. And we're about to launch the Delta Science Tracker, which will be a web-based tool that tracks all science activities in the Delta. It's searchable by topic area, by agency providing the funding, and it will provide a link to the data and the products, including publications. So I promised that I would highlight our ongoing working group that is partnering with NCs, and that working group is focused on understanding drivers of the estuarine food supply. Some of you might be aware that the Delta is one of the most imperiled ecosystems in the state. And one of the ways in which that imperilment has manifested itself is that in the 1990s, many of our native fish species underwent almost a step decline, which has been referred to as pelagic organism decline. There's no smoking gun associated with a known driver or a single known driver of that decline. Rather, it's understood that many interacting factors have driven that decline in native fish species. And it's likely that one of those factors is related to food web restrictions. However, we don't have a good understanding of what controls the basis of the food web and what the primary drivers of the food web are. And so this group really set out to tackle that big question. And they broke this into three main questions. In addition to what the key drivers are, they've also looked at how have invasive species shaped Delta communities. The Delta is one of the world's most invaded ecosystems. And then three, how can restoration and flow management protect and enhance the food supply for fishes of management interest under future climate and management scenarios? This might be of interest to you all because there's a really nice resource that contains all products and training available via this website that you're looking at. And this next slide is really busy, but it just gives you a sense for the breadth and depth of those resources that are available, including code resources, code packages, and really nice integrated data sets. And I'm just going to return to one of the data sets that Sam had shown, which is the zooplankton data set. There are still so many questions regarding zooplankton and the role that it plays in Delta food webs, or even what trends in the zooplankton community have been. And so I think if there are groups looking for a project, the data contained within this Shiny app could be a really good starting point to address the low-hanging fruit opportunity of really what are these trends, what are the trends behind zooplankton data, and what are some of the drivers. I promised I would also show a slide very similar to what Sam presented, just again, putting a plug in for the Delta Science programs, our Shiny app website. In addition to the zooplankton synthesis, you'll find data on salmon telemetry. You'll find continuous water data, temperature, and I'll return to that and tell a little bit of a story around that at the end of the talk. And then the other thing that I wanted to highlight is perhaps particularly relevant to topical challenge that you all are facing as part of this symposium is an app that maps out patterns of social vulnerability within the Delta. So what's being shown right now is just the percentage of households with an income substantially below the federal poverty line, but there are a number of other indicators that you could toggle to and combine with environmental data sets or other data sets. So just to pivot a little bit, another thing that is one of our core functions is to fund science and to coordinate with a number of agencies performing science. And in service of both of these missions, we put out what's known as the Science Action Agenda, which is a 4 to 5 year visioning document that prioritizes science investments in topical areas that are best poised to make an impact for management. And so we just put out the 2022 to 2026 Science Action Agenda in April. It was the culmination of a nearly 2-year process of extensive co-production and engagement with a number of different stakeholders from the management community and academic community. And in that document, you'll find 25 priority science actions. I should say that we put out biennial funding solicitations for large projects, as well as biennial solicitations for Delta Science Fellows that are available to master's students, PhD students, and postdocs. All those opportunities need to address management needs and science actions within the Science Action Agenda. And so I just wanted to highlight a few for you today that show the priority that we are placing on synthesis and some of the topics that are in great need of synthesis. And one of those is invasive species. There's a call to synthesize existing knowledge and conduct interdisciplinary research to evaluate costs and benefits of strategies for minimizing the introduction and spread of invasive species. One that's particularly relevant to the challenge that you all are facing is one focused on human communities. And I think Emily's data set kind of falls into the category of addressing this need. So synthesize existing data and collaboratively develop long-term data collection and monitoring strategies to address knowledge gaps on human communities within the Delta, with the goal of tracking and modeling metrics of resilience, equity, and well-being over time. And then another priority action related to synthesis is synthesizing data on contaminants. I think I've heard there are something like 22 different databases of contaminants for the Bay Delta. And so there's a need to quantify spatial and temporal patterns and trends of chemical contaminants broadly, and then to evaluate ecosystem effects of those contaminants. So just delving into the human component and the need for synthesis of human data a little bit further, I wanted to highlight that there is a social science community of practice that was recently convened, and they are planning an event in 2022, sometime this fall, that will consist of training and a workshop. And that training is going to focus on educating the public on social science disciplines and particular types of integrated projects. How can we integrate social science objectives with biophysical science objectives? And then a week later, that will be followed up by a workshop in which the vision is to develop collaborative working teams to identify and perform social science syntheses. And so I've asked Dr. Jessica Rudnick, who's running the social science community of practice, for some of the low-hanging fruit ideas that are out there right now. And one might be the opportunity to link environmental monitoring data to human health data. And so this website that is being shown, OEHHA, provides spatially explicit health data with respect to several different types of indices. And that could be coupled with environmental data from a number of the other databases that you've seen here today. So harmful algal blooms and synthesizing information about harmful algal blooms is another priority that is prominent in the Science Action Agenda. And fortunately, Ali showed you earlier the harmful algal blooms incidence reports map, which is an excellent resource provided by the State Water Resources Control Board. Harmful algal blooms, admittedly, is something that has only become a recent need for monitoring. There were virtually no HABs events in the Delta about 10 or so years ago. And so the community is really figuring out how to spin up a robust monitoring effort around harmful algal blooms and understand what the drivers of particular HABs events are at particular locations and times. And so there's a workshop that will be taking place in November to bring the community together around this topic, identify opportunities for synthesis, but also outline a strategy for doing more robust monitoring. And so related to that, in just kind of brainstorming what some of the low-hanging fruit opportunities are, I think it could be very useful if project teams associated with the Water Data Challenge were interested in diving a little deeper into the HABs problem in the Delta. And I think the HABs incidence reports map is a really good starting point. I think there are opportunities to extract data from the geographic area of the Delta and start developing time series or scrape the database for text associated with known types of organisms. I just wanted to show you what the actual database looks like, and there's a lot of really good information here. So I am nearly out of time here. I just wanted to skip ahead to showcasing what could be done, to showcasing like a really excellent example of what could be done with the sort of synthesis that we're calling for. And so I'm actually going to highlight some work that Sam Bashevkin was highly involved in for the Delta Science program on synthesizing temperature dynamics. Many listed species in the Delta are at the southern extent of their range, and so they're very sensitive to increases in temperature. And some low-hanging fruit that Sam and team identified was to identify what the temperature trends have been in the Delta and what regions are experiencing warming at a greater rate than others. And so based on IEP, based on over 5 decades of IEP temperature data, they fit models to the Delta and identified that there is a substantial warming trend over the past 50 years that's most pronounced in the North Delta. They also, in a separate paper, looked at associations between inflows and temperatures. This begins to get at the question of maybe the extent to which we could control temperature by modulating inflows, although this study did not look at causality, it only looked at correlation. But there was a substantial negative temperature to inflow relationship that they uncovered through this analysis, where during periods of higher inflow, water temperatures tend to be cooler. So this is some really important foundational research. Some other research by this group looked at thermal stratification, which can provide temperature refugia near the bed for native species. And what they found is that the thermal landscapes that are perhaps suitable for native species like Chinook salmon and Delta smelt here are being shown in yellow and purple. And they looked at changes in these ranges, these spatial locations over different temperature anomalies. And this is something that could be really informative for where we might have successful releases of captive populations. And then coming soon, this paper is now in review, was a related study looking at the number of days that are thermally stressful for a suite of native species through the Delta. And that was a very sobering paper. It found that thermal stress is already quite common, and stressful days may increase by up to 30 days from an average year to a warm year. And so this is something that we really need to be paying close attention to and coming up with adaptation strategies for native species. So with that, I will close my presentation and ask if there are any questions, and I'm happy to answer emails and point you to these resources and some of the management priorities more directly if they're of interest.
Hi, Laurel. We do have one question so far. You mentioned social vulnerability indicators. How is your data set different from that of Callan virus screen?
I don't know the answer to that question. That's a great question, and I will try to find out.
Okay, and with that, we don't have any other questions that have come up.
I have one. So given your role as both a professor and a person who represents a lead scientist at a state agency, what do you see the future few years looking like in terms of pipelines and opportunities for creating better data science, water data science, collaboratives, workforces?
That's a great question, Greg, and one that we've been thinking about a lot. If you talk to people like Sam and others at the State Water Resources Control Board who are involved in synthesis, I think there's widespread agreement that there is that the need for synthesis work greatly outpaces the bodies and minds that agencies tend to vote to synthesis right now. I also know from talking to folks at different agencies and within the water contractors that they're having a hard time hiring people into positions right now with the needed skills. And so I think events such as this one are a really great way to open up lines of communication that could feed into the pipeline that you speak about. I'm very much engaged with the Berkeley Institute of Data Sciences, and their new data science division has a data discovery capstone project that I think is a program that does a really great job of matching up teams of students with mentors in industry or in government to tackle projects, maybe projects like the project opportunities that I described here. And so I think we need to see more and more of that type of relationship building between academia and government. And I think the future is very bright here. I only see more and more of these opportunities being developed as time goes on.
That's great. Thank you.
And I would say that just to put another plug in for our funding programs at the Delta Science Program, we do tend to build long-lasting relationships with the fellows that we fund and with the lab group teams that we fund who are working on these projects that advance the science action agenda. And so that's one way that we grow our network. And our Delta Science Fellowship Program has a community mentor aspect to it, where we help prospective fellows identify community mentors who regularly apply that science in service of meeting management or advocacy objectives. So, for those of you who might not have heard of that, the deadline, unfortunately, for this round has just recently passed, but please do keep that on your radar and help us spread the word about these opportunities. And we're really emphasizing social science projects in both of those funding programs.
Yeah, thanks. And that actually leads to like the final thought, and actually I'll share your thoughts because we have a minute still. The pipeline that we've seen in the past has been, driven by the forces of, I'll just say it, racist systems that have been put in place in the past decade. So when you were thinking about a more equitable workforce pipeline process, do you have any special thoughts? I know Berkeley is probably a pretty good source, but we have a lot of universities, community colleges, and other places to think about this.
Yeah, so one of the things that we did last year, and it was kind of serendipitous how this came about, but a contact that I have at Jewish Vocational Services, which is actually a non-religious nonprofit, runs a data science boot camp for people looking to make a career transition. And these are all often folks from underrepresented communities. And she was looking, she had noticed that a lot of people in that cohort were really interested in this question of data science for the greater good. And she was looking to match them with capstone experiences in government or nonprofit. And I think, I think there's a lot of opportunities for really energetic people and government to take the initiative and make these internship programs happen. I think if we can pay people adequately in these roles as interns, which can sometimes be difficult in a government agency, But I think, you know, compensating people for their service in these internships is really an important factor in making these opportunities more equitable. So there's a lot of these opportunities to form partnerships. I think if you take the initiative and look for them, there's a lot that you can do to make these partnerships happen.
That's great. Thank you for that. And thank you for your time today, sharing all your thoughts and knowledge and expertise. Next, we have a speaker from the Cali PA family, Kamal Bangia. I hope I got that right or close. I apologize if I didn't. Works for the Office of Environmental Health Hazard Assessment, is here to talk about an amazing project called CalEnviroScreen. And you can actually field that question if you know anything about the Bay Delta's social vulnerability versus CalEnviroScreen at the end of your talk if you want. But take it away.
Thank you. I am just getting through some technical difficulties, but there we go. Yeah, it works. Great. So yeah, hi everyone. I'm Komal Bangia, and I'm a research scientist at the Office of Environmental Health Hazard Assessment. And so what is OEHA? What do we do? We have a lot of different programs under that that we work on. We have CalEnviroScreen, we have Biomonitoring California, we work on Proposition 65, we have a climate change research group, we do risk assessments and risk method development. So we have a lot of different programs, and I specifically work on CalEnviroScreen. And so what is CalEnviroScreen? CalEnviroScreen is an environmental justice screening method and tool. It's a cumulative analysis of relative burdens in California communities from pollution and pollution vulnerability. So in CalEnviroScreen 4.0, we had 21 indicators, and they're combined into one single score at the census tract level. There's about 8,000 census tracts in California. So that's what we mean by communities when we talk about communities in California. So the goal of CalEnviroScreen is to identify California communities that are most affected by multiple sources of pollution, and also communities that are especially vulnerable to the effects of pollution. And the most recent version was released in October 2021. And so here we have the 4 components of CalEnviroScreen. We have the exposures component, environmental effects, sensitive populations, and socioeconomic factors. I won't go into too much detail about this because I want to get to our drinking water indicator, but I do want to mention that the drinking water indicators in the exposures component. And then also the new indicator that we added in 4.0 is the children's-led risk from housing indicator, which is also in the exposures component. And so here's sort of the model that we have to combine the indicators and get a score by tract. We average all the indicators within each component, and then we average the components, and then we multiply pollution burden and population characteristics to get a score for each tract. And it's all ranked into percentiles in the end. All right, so for CalEnviroScreen 4.0, I want to briefly mention all our major updates in that that version. So overall, all the indicators, we've updated the dates of the dates that we dates of the data. But these are the major updates. For one, we updated the contaminants, and we have a more complete set of service boundaries for our drinking water indicator, and I'll talk about that more in a bit. For our pesticide use indicator, we added additional pesticides for that indicator. We added dairies and feedlots to the groundwater threats indicator. We added chrome metal plating facilities to the hazardous waste indicator. And then for PM 2.5, so the satellite data used to be at the 10-kilometer grid, but now it's a 1-kilometer grid. For diesel, the emissions used to be at a 4-kilometer grid, now it's 1-kilometer grid. So it's fine-tuning those indicators as well. And then the last thing is that we incorporate better data for indicators such as traffic and PM and toxic releases at the California-Mexico border. And so our CalEnviroScreen 4.0 results are online. We have it in an Excel format. We have it in a geodatabase, and we also have the online maps to look at. Here you can see that this image of California highlights the areas of the state that have the highest burdens and population vulnerability. You see a lot of San Joaquin Valley and areas of Los Angeles, like the greater Los Angeles area as well, being highlighted there. And then one of the major uses of CalEnviroScreen, the main use really is to designate disadvantaged communities for investments from greenhouse gas reduction funds. And here you see a map of the LA area and the disadvantaged communities in that area. All right, so now I'll talk about our drinking water indicator. So this is a little bit different, from we're talking about more the quality of the water at the tap, as close to the tap as possible, not necessarily at the source of water. And this indicator's in CalEnviroScreen as one of the indicators. So again, what we're trying to do here is we're trying to get at water quality for what people are drinking. We combine multiple contaminants into one index, and we compare those from track to track. And what we do is we convert data that's at the water system level, and then also we capture areas outside of water systems, and we convert all that to the census tract level. We start out by taking advantage of water system service boundaries. So in 4.0, when we put that together, we got the boundaries from the water boundary tool that was at Tracking California at the time. We use water quality monitoring data from the drinking water program at the water board. And then for areas outside of the water systems, we summarize the ambient water quality data by public land survey system section. So that's a 1 by 1 mile grid. And we use data from the groundwater ambient monitoring and assessment program. And so we put all this together, we put all the boundary stuff together by first, taking the water boundary tool, we clean up the boundaries, we remove overlays so that there's one clean layer. And then we add in the areas outside of those service areas, and we combine that all into one layer of systems and sections. The townships I have townships on there, but that was for 3.0, but now we've improved it to go down to the section level. And so how do we put all that water quality data together? We get averages, or we calculate the averages by water system, and then we also get the averages by section. And then we take those averages and we population weight them to the census tract level. And then we rank each contaminant and sum them across to get one score for each tract. And so it's a little bit complicated, but that's sort of the general process. And in CalEnviroScreen 4.0, we added 3 new contaminants. We added ethylene dibromide, gross alpha, and haloacetic acids. And we also used data from the lead and copper rule, the lead data from the lead and copper rule. And then we've always included MCL violations and TCR violations, but this time around, we also included action level exceedances and included that into the MCL violations. And so actually, before I do this, I wanted to go and just kind of show you the results. I was a little bit mistaken. I thought I was going to be able to do a demo, but I know I'm not able to do that. But this is a small map of the drinking water indicator. I do want to mention, what we did see was, of course, areas of San Joaquin Valley, greater areas of LA. Those areas stood out. I do want to encourage you guys to go to our drinking water indicator map. So all our indicator maps are online, and you can, in the drinking water indicator, you can click on a tract and see the individual contaminant percentile scores as well. So you can see, if lead is high in an area, or if arsenic is high in an area, or in a census tract. So there's a lot of information there. If you go to the individual indicator maps. So go back, because I wanted to talk a little bit about some of the data gaps that we were faced with, and then how we sort of addressed them in this indicator. So for one, since 2.0, the service boundaries weren't complete, but working over time with Tracking California, we were able to in 4.0 get a complete set of boundaries. For the most part, I think there were like only 10 that we didn't have. But and then the other gap, or the other challenge that we have is that water systems in general, the sampling frequency varies by system size and where the system's located. And to address this, we use 9 years of data to get as much data as possible, enough samples as possible for contaminants that are highly tested and detected. And so those are some of the criteria that we have for the contaminant selection process. And then another data gap is, for tribal water systems, there's a lack of reported data. For 4.0, we were able to find that 12 of the tribal water systems, they purchased their water from wholesale systems. And so we incorporated that wholesale system data for those tribal water systems. And we're hoping to work on this more in the future, to learn more about tribal water system, water quality data, and see if there's anything that we can incorporate. So the next data gap is about uniformity within water system boundaries. So, currently we have this assumption that within a water system boundary, all the water quality is uniform. We kind of have to make that assumption because it's very hard to get like the information for within water systems. But I think, for LA Water and Power, we were able to divide that into the subsections early on because they have that data readily available, or that information readily available. But one of the goals is in the future to look at other larger water systems and to be able to understand which sample points are more relevant to certain areas of those water systems. So, and then the last thing that I want to talk about, there's many data gaps, but the last thing I'm going to talk about here is for domestic well users, it's always a challenge to understand what domestic well users are drinking and what their water quality is like. We decided to summarize it by the section level, and by doing this, we did not need to know the exact location of domestic wells, which is another data gap. But yeah, so that's how we were able to kind of understand areas outside of water systems. Let's see. Okay, so the uses of the drinking water indicator, I'll kind of go through this a little bit quicker because of time. But so besides being incorporated into CalEnviroScreen, our drinking water indicator data was also used in the water quality component of our Human Right to Water report and tool, which was released in January 2021. And that tool, you're able to see water quality, you're able to see water accessibility and water affordability components. The cool thing that the water quality data enabled us to do was to do some analyses on social equity. And so here you can see that higher water quality scores in severely disadvantaged communities and disadvantaged communities. It was higher than in non-disadvantaged communities. So that was kind of interesting. And then another use is, I work with researchers a lot to help with, water quality data in California. And along with that, we also published a study on the assessment of contaminants in California drinking water, and we assessed regional differences along with system size differences. And here we found information on on domestic well users. And for example, in Northern California, there seemed to be a disproportional burden for DBCP there. But you can also get an idea, about community water systems, and like for example, nitrate seem to be disproportionately high in San Joaquin Valley. So we were able to understand contaminant by contaminant in different areas of California. We also did a trend test. So we were able to find that, this the domestic well users were had arsenic and cadmium average, which was higher than small water systems even, and the trend even lower or went negatively as you go from small water systems to large water systems. And then for like industrial contaminants, the larger water systems were disproportionately more burdened. So that was very interesting too. So overall, the next steps for our drinking water indicator, we want to, again, get more precise selection of sampling for areas within water systems. We also want to understand state small water systems more. I think state small water systems are a huge challenge because we can't necessarily make the assumption that we do with domestic wells because they're an actual water system, but they're also not, regulated by the state. So they're a big challenge. And then also we want to explore emerging contaminants like PFAS. The other thing I want to mention about the Human Right to Water report is that it does allow for a baseline for future assessment for trends over time. So that could be a possibility in the future. And then the last thing, so these days, instead of just focusing on water quality, we're all trying to focus on the human right to water. So accessibility and affordability. And OEHA is currently working with the Water Board in the SAFER program to help identify affordability metrics for domestic well users and state small water systems. So that's exciting and something that we're continuing to work on with them. And then I just wanted to show you guys, I thought I was going to do a demo, but you guys are going to get all the websites for all our tools. And I hope that you guys explore them. Even our Human Right to Water data tool is really interesting. You can get the scores by water quality, accessibility, and affordability, and the indicators within each component. And then, I just wanted to share my email address and our Calvin Virus Screen email address and Human Right to Water email address. Feel free to send me any questions you have here. Yeah, thank you.
That's great. Thank you. Bev, do we have questions?
Yeah, we do have one question. Before I ask this, I just want to point out, if you haven't been paying attention to the chat, that Anna has been madly putting in links to everything that people are talking about. So there's a ton of resources there in that chat. And then to the question, for Calvin Virus Screen 4.0, when you look at MCL violations, do you just look to see if it exceeds the MCL, or do you filter out any sources that may have done some treatment?
So we actually get the information from the Water Board if they've received an MCL violation. So that, their criteria on receiving MCL violations, I think, varies by contaminant. So our MCL violation measure is a sum of any contaminant MCL. So if they've gotten an MCL for any contaminant between the years of 2011 to 2019, we add it in there. And so before we get to the census tract level by water system, we have a total violations they've gotten in those years. And then for 4.0, we've added in the action level exceedances. So if they've gotten an action level exceedance as well, we've added it into that.
Okay, thank you. That's the only question that we've had come in.
I have one more. I'm wondering, are you considering in the future, like 5.0 or whatever, looking at maybe lead in the distribution pipelines or any other kinds of like drinking water health risks that would inform your tool?
Yeah, so I mean, one comment that we've gotten is to include service lines, like lead service lines. And so that, I believe it's not complete, but I'm happy to learn more about it. I believe it's not complete yet, but we were thinking about at least adding it as a layer to look at in our drinking water indicator. And we've done that before with other indicators. We've added monitors for PM, and we've done that where we can add other layers just so people can look. So I think the service lines might be a good idea to add in there. But we haven't looked at anything else, but we're happy to get any kind of suggestions to add in there.
Cool.
Yeah, so far for contaminants, I've been given the suggestion to look at PFAS, which we looked at when we were putting together 4.0, and at the time there wasn't very much data on PFAS, but I believe at this point there's probably a lot more, and so we'll definitely be looking at that.
Yeah, and there's brand new threshold information coming out that's very, very low. So yeah, the only other thought that comes to mind is maybe in the future microplastics, if that continues to be something that's monitored at drinking water locations. Cool.
Yeah, definitely keep that in mind. Thank you.
Thank you. Thank you for your time and for sharing your expertise and your presentation. That concludes the panel of speakers we have for today. In terms of the rest of the data symposium and challenge, that also concludes the symposium portion. So I want to give a huge shout out to Anna Holder for pulling this off again. And the rest of the team, thank you so much for your help. We've had lots of helpers. We have one more event. We have a workshop in the morning for the Water Data Challenge. I encourage you, even if you're, as I mentioned earlier, sitting on a fence of, should I go to one of these things? I'm not sure. I don't really work with data. Whatever your feelings, go ahead and come on in. It's a very inclusive place, and you'll find that the conversations are almost always very interesting. And so I encourage you to do that. Make sure I'm looking at my chats. And I do think there's a lot of opportunity for the topics that we heard about in the presentations today and yesterday morning to turn into projects. And so I encourage folks out there in either YouTube or webinar land to think about that. There are going to be some commitment events coming up. We hope to have a date and a location pretty soon for a regular partner that's helped us out in the past with commitment events. The UC Davis instructor in charge of their data science center there. And so that might be one. We also hope to link some other commitment events to other activities happening this summer. And if you have an idea, or if you'd like to explore what it means to hold a commitment event, please reach out to us. All that information is on the website for waterchallenge.data.ca.gov. We have a Slack channel that's growing with content for people that are sharing their ideas. And I want to thank you again. And Anna, did you want to say something?
Yeah, so I guess that before we sign off for today, we wanted to make folks aware of other things happening this summer. We kind of talked a little bit about our mascot for this year, the salmon, and we're going to try and put something together where folks can kind of spawn more conversation about why salmon are important to them and just keep kind of the community momentum going. So we will have details for that kind of out on our email list that I'll show in the next couple slides. But we've mentioned throughout the symposium that we partner with a lot of folks, and they're also doing really great things. So we mentioned the Water Data Challenge, to partnering with the West Big Data Innovation Hub, among many others. So again, reiterating what Greg said, if you want to host or attend a commitment event, please stay tuned. If you're one of the teams that have been forming during our workshops over the past couple of days, the deadline to submit projects will be October 15th. And again, we'll keep updates flowing through email and Slack and things like that. But we also have a close partner with the California Data Collaborative, and they have their own summit happening in August in UC Irvine. And we wanted to kind of shout out to them and share their webpage and information. So the flavor of the conversations are a little bit different, but still awesome. And if you want to learn more, the website's in the chat. We also have Ali, Dunn, and Tessa Fojit mentioned earlier today the bioassessment, the swamp bioassessment monitoring program, and we actually have an annual meeting for the California Aquatic Bioassessment Work Group, or CABW, in partnership with the California Society for Freshwater Science. That happens in October every year, and it's happening this year, and it's going to be in person, assuming it's safe to do so. So that's October 11th and 12th. And more information on that will be provided on the website at that bit.ly link. If you're interested in being a presenter or presenting a poster, the deadline for submission is July 30th, so a month from today. And the super long and ugly Google form link that I put in the chat is a link to that form. And then registration will be open for quite some time, but that is through Eventbrite. And the link is in the chat there. Bev highlighted that I have been adding a lot of links in the chat over the past couple of days. I'm also creating a document so that we'll send out later this week or early next week that has links to these recordings, as well as all of the resources that our amazing presenters have been sharing with us. So stay tuned for that. And we'll be sharing that resource directly to you who have been able to register for the event through your emails. But if you are watching us on YouTube, we recommend staying informed through our College of Water Informatics and just joining that email list. And so you can stay up to date on what's happening with the challenge, other exciting opportunities, and opportunities to connect with the community pop up. And I think that's it. So again, Greg, feel free to jump in, but I just wanted to reiterate deep gratitude for all of the presenters and panelists and keynotes and our board chair for saying yes to participating and engaging in this event. All of the folks at the workshops who've attended the last couple of days, and you watching now. We know it's draining to stay on a Zoom webinar or a Zoom meeting for hours and hours and hours. And we thank you all so much for being part of this community and dedicating that space for us to share exciting things with you.
Happy Pride Month.
Yeah.
And last year we had the honor of having an amazing keynote presenter that helped expand our awareness of how to raise the vulnerable communities up through that. And then one more question, how should environmental scientists bring up their interest in participating, initiating a project like those brought up here with our supervisors? Would this not lead to conflicts with our normal job duties and responsibilities? I'll take a stab at that. Anna, if you want to take a stab at that too.
Nope, all you.
Well, I think, for sure, if you're going to try to do this through your work channel, like on your work time, talk to your supervisor and find out, are there boundaries and limits to both the work that you're doing and the time. But I think many of the projects, depending on your role, like there is a role for, I don't know, I think you're a Department of Water Resources employee, but I'm not 100% sure. But if the teams are looking for someone who can help inform and put context into their project, that seems like it's a role that does not create any conflict other than the potential time part. If the project's going to be a pointy-ended thing that sort of says state agencies should be doing something better and different because of this amazing data insight that we're looking at, that's probably something that you want to consider on your own and seek some advice. I have come up with a couple of instances in my career where conflict of interest was real, and I got advice and I avoided it. So I would just encourage you to sort of be thoughtful about the details. And if you want to reach out and talk more, feel free to contact me.
Cool. Well, thanks everyone. Enjoy the rest of your afternoon or evening, depending on, or morning, depending on where you are in the world. And we'll close and see you all soon. So thanks so much.